Here we are at our last section for Chapter 2. This is Section 4, Chemical Reactions and Enzymes. Our key concepts that we're dealing with are what happens to chemical bonds during chemical reactions, how do energy changes affect whether a chemical reaction will occur, why are enzymes important to living things? And out of all of these, that last bullet is the one that is a theme that we'll carry out throughout the year. So when we talk about chemical reactions, we talk about the breaking and forming of bonds between atoms and molecules. Chemical reactions will change substances into different ones by breaking and forming different chemical bonds. The reactants are changed during a chemical reaction. So reactants are what we start out with. They're the ingredients for whatever it is that you're making. And the products are made, they're produced by the chemical reaction. So that's what you end up with. So if you're baking a cake, for example, your reactants are the ingredients like the eggs, flour, oil, milk, butter, whatever. And then the product is the finished cake. So think of a chemical reaction like that. Here we have reactant. The arrow, if you were to put a word in instead of the arrow means produces or synthesizes or makes. That's what the arrow means when you see it written. And we'll actually be looking at different chemical reactions during the year, and you'll have to understand that that's what the arrow means. Please make sure you are highlighting. So bond energy is the amount of energy that breaks a bond. It takes a certain amount of energy to pull these atoms and molecules apart. If you remember, we talked about covalent bonds and ionic bonds, and those are pretty strong bonds. We also have hydrogen bonds that are pretty strong attraction, and these places where atoms and molecules are stuck together, it takes energy to pull them apart. And energy does get released when a bond forms between atoms or molecules. A reaction will be at equilibrium when the reactants and products are forming at the same rate. So here's an example. When we have carbon dioxide in our bodies, it actually has to form a compound with water, and that way it can be transported through your blood, and then you can exhale it at your lungs. And so what we see here is the reaction that occurs after your body produces carbon dioxide. It reacts with water in your body, and it forms carbonic acid so that it can travel in your blood, and then you can, at your lungs, it actually goes back to carbon dioxide and water, and you exhale that. And if you pay attention when you exhale, if you were to breathe on a mirror, for example, you actually form condensation, and that's the water. Chemical reactions release or absorb energy. And activation energy is the amount of energy that needs to be absorbed to start a reaction. Any chemical reaction requires at least a little bit of energy input to get things going. And so you can think of activation energy like a hill. It takes certain reactions a lot of energy. Some reactions it doesn't require very much. But that hill, for example, is the activation energy. You find the reactants before that hill, and the products are going to be after that hill. An exothermic reaction is going to release more energy than it absorbs. And if it were occurring inside of a flask, but like in the pictures that you see here, and you were to touch it, that flask would feel warm. So an exothermic reaction releases more energy and it'll feel warm. The reactants will have higher bond energies than the products. Excess energy is released by the reaction. And here they're just showing you at the end, the products level of energy is going to be lower than the reactants. Endothermic reactions absorb more energy than they release. The reactants will have lower bond energies than the products. And these reactions are going to feel cold if you were able to touch them. Energy is absorbed by the reaction to make up the difference. So here you see the product's energy level is going to be higher than the reactants. And honestly, the activation energies and endothermic, exothermic, those are all things that I'm explaining so that you can understand what a catalyst is and why that's important. So out of all of this, understanding what a catalyst is and an enzyme that we'll talk about shortly, that is the important part I want you to pull out of all of these chemical discussions. So a catalyst is going to lower the activation energy. It's going to make that hill smaller. The catalysts are substances that speed up chemical reactions. They decrease the activation energy, and they can increase the reaction rate. So here you can see the solid orange line is what the regular activation energy would be, and then the dotted line is what it is for a reaction that has a catalyst added to it. So it makes it easier for this reaction to occur. Enzymes are types of catalysts. Enzymes are biological catalysts. Enzymes are catalysts that are going to be found inside our bodies, and they allow chemical reactions to occur under very controlled conditions. So enzymes, like I said, are catalysts in living things. They are needed for almost every single process that occurs in your body. If you did not have enzymes inside your body, you wouldn't be alive. Most enzymes are proteins, and this is something I don't believe I have you highlighting, but I think you should. This is going to come up multiple times during the year, and you need to know this particular phrase, that enzymes are proteins, and they're very specific. Their names usually end in ASE. If you recall before, I told you sugars end in OSE, but enzyme names are going to end in ASE. And this will be important for you to know, because if you encounter a question that is talking about all these weird names, if you look at the ending and it ends in ASE, you know that they're talking about an enzyme. You don't have to know the name of the enzyme in most cases. You just have to know, oh, that's an enzyme, that means it's a protein, and then you can go on from there. Here's some example names: lactase, polymerase, ligase, and you're going to see these names again, but they all end with ASE, and they are types of enzymes, and usually they're named for what they do. Lactase actually breaks down the sugar lactose. Polymerase helps to build polymers. So please make sure you highlight enzymes, very important term. Disruptions in homeostasis can prevent enzymes from functioning. Enzymes function best in a small range of conditions. Change in temperature and pH can break hydrogen bonds that are in these proteins, these enzymes. And an enzyme's function depends on its structure. And this is a very key point that will come up again and again, is that an enzyme can only function if it has the right shape. Usually enzymes have only one specific thing that they can do, and they have to do it, or they can only do it if they are in the certain shape that they're supposed to be. An enzyme structure allows only certain reactants to bind to the enzyme. These are called substrates. The specific thing that an enzyme works on is called a substrate. And they kind of fit together, enzymes with substrates, like a puzzle. And the place where the substrate fits into an enzyme is called the active site. And this diagram is showing you the big giant purple thing is the enzyme. It has a very specific shape. And then you can see the blue and the orange puzzle pieces, those are the reactants or the substrate, and they fit in certain places on the enzyme. And that's the active site. The enzyme typically will not change shape during a reaction. It'll just help these substrates come together and change their bonds and form a reactant or form a product. Please highlight substrates. So the lock and key model helps illustrate how enzymes function. If you think about how you have a lock on your door to your home and only a certain shape of a key will fit that lock, that's kind of how enzymes and substrates work. So substrates are brought together with an enzyme. The bonds in the substrates are weakened. They are changed, and the catalyzed reaction can then form a product, and then that product gets released from the enzyme. Enzymes in the human body will work the best at 98.6 degrees Fahrenheit or 37 degrees Celsius. When you get a fever, the doctors get worried when it gets too high, like 104, 105, because then your enzymes in your body stop working the way that they're supposed to. They start to fall apart. They lose their shapes. And that's a problem. You will die if your enzymes don't work. So if the temperature goes too high, the bonds in the enzyme will begin to break or denature. And if your body temperature goes too low, they don't necessarily denature, they just stop working. Denature is a term that you're going to need to remember, so make sure you highlight that, please. The ideal pH level, the acid-base level in your body for most of the enzymes is between 6.5 and 7.5, except in your stomach and your intestines, the pH is going to be much different, and those enzymes work better when the pH is very low. But if your pH goes too high or too low, the bonds in the enzymes will break or denature, and then the enzymes change shape. If they change shape, they don't do what they're supposed to do. The shape of the enzyme is very important because it has to fit exactly to the substrate like a lock and key. Other things that can affect the rate of a reaction is the enzyme concentration. How much of that enzyme is available will determine how fast reactions can occur. So an increase in the amount of enzyme usually equals an increase of how fast the reaction happens. More enzymes means they are more frequently going to collide with the substrate. Also, substrate concentration, if there is more substrate to work with, more of the ingredients or reactants. An increase in the amount of substrate usually increases how fast the reaction happens. If there's more substrate, they are more frequently going to collide with the enzyme. So this last slide is probably the most important slide out of this entire slideshow, because for proteins, it's the shape that matters. They do their job because of their shape. And remember, enzymes are proteins. If they have the wrong shape, they cannot do what they're supposed to do. So if you unfold a protein, it destroys its shape. If it doesn't have the right shape, it can't do its job. Unfolding proteins is called denaturing. And temperature changes can denature enzymes or proteins, and changing pH levels can denature proteins or unfold them. That takes us to the end of our Chapter 2-4 notes that talks about enzymes and chemical reactions. Please make sure you filled in all of your notes in the Chapter 2 note packet, and now you should go take the quiz on Section 4. Good luck.